and now we're gonna hear something some about therapeutic measures. Thank you, Dr. Eftekharpour. The stage is yours.
Hello, everyone. I hope you guys can see me and can hear me.
Yes, we can see and hear you
Great,
perfectly.
great. First of all, a good time wherever you are. It's a beautiful morning here in Winnipeg, Manitoba, Canada. And I'm going to be talking about some new findings in my lab. These data are not published, and this is my first time at UCERN, I like to treat this as a learning, conference and trying to teach hopefully new students. So I have some learning objective here for you. I'm going to talk about pathophysiology of Alzheimer's disease, uh, review quickly the treatments. The most important thing that I want you to take from this is that we are now learning that Alzheimer's disease is an acquired laminopathy. And what is laminopathy is damage to nuclear lamina. We will be talking about the structure and function of the nuclear lamina, and we will bring examples from human cell lines and animals to prove our points. And, based on what I'm going to be talking, we will be talking about new potential therapies that hopefully will arise from research like this. Today, I want to also acknowledge that is Remembrance Day here in Canada, and the poppies here are for us to remember those who fought for their countries all over the world. So someone in the world, every 3 seconds, one person in the world will be diagnosed with Alzheimer's disease. Under the microscope, you will see this plaque in their brain. This would be amyloid plaque. If you use, and they are containing amyloid beta 42, a very sticky material that sits outside the neurons. If you stain them for staining for phosphorylated tau protein, you will also see them inside the neurons. These will cause a degeneration of the neurons, and they will be acquiring some foreign bodies inside their cells, and they will be bodies. One of the most important thing, in my opinion, contributors is the oxidative stress, and this is evidenced by loss of antioxidants in the brain tissue. These all causes progressive neuronal loss, and eventually they will lead to cortical atrophy and change in mood and behavior that we know from Alzheimer's disease. There are certainly some treatments, limited treatments for Alzheimer's disease patient. Non-pharmacological treatments are mostly behavioral therapy. These are just is given to calm the patient down and in the earlier stages to inform them of what's going to happen. There are some reports of effectiveness of things like music therapy or aromatherapy and psychotherapies. When it gets to pharmacological treatments, things are not as good as we would hope. There are two groups of drugs that are needed. The drugs that are currently available, these are symptomatic therapies. They only look at the symptoms and try to alleviate those symptoms. These are two big groups. One is acetylcholinesterase inhibitors. The other groups are the NMDA antagonists, and they are given just basically to delay the onset of the disease. There is no disease-modifying treatments, and there is no FDA approved. There's currently no FDA-approved drug that can actually prevent or reverse the disease. So this is an ongoing search. There is hope in future. There are more than 150 clinical trials at different phases that are undergoing. Strategies that to reduce amyloid beta by different inhibitors are ongoing. Reduction of amyloid beta aggregation using special peptides is a possibility. We still don't know about it. You could use immunotherapy against amyloid beta or anti-tau, and these are all things that are going. As of 2 days ago, this was a drug from Biogen, was in clinical trials, and it is awaiting FDA approval. This is, unfortunately, I do not think that it will receive approval. If you have a stock in that company, there are, there are not enough data to support the efficacy of this drug. And for your information, there has not been any new drug since 2002 approved by FDA for Alzheimer's disease. So there is definitely a need, and there is something that needs to be changed about how we treat Alzheimer's disease and how we look at it. So a new aspect of Alzheimer's disease has come out, and it's called laminopathy. And laminopathy is damage to nuclear lamina. For those of you like me that did your cell biology last century, you wouldn't know nuclear lamina. This is what my situation. We didn't know about nuclear lamina, but this is the nuclear envelope here. Under the nuclear envelope, you have a thin layer of protein. This is the nuclear lamina. Nuclear lamina binds to nuclear envelope, and on the other side, it also binds to chromatin. And as you would imagine, this would have to regulate protein and gene regulation at a certain degree. We know that in Alzheimer's disease, there is damage to nuclear lamina. And here are some of our own human tissue that we have acquired, and we have stained them. You are looking at lamin B1. This is a protein that is located under the nuclear envelope. And when we stain it, you can see these beautiful round cell. These are neurons as specified by neuronal NUN. And they are perfect, almost no damage we can see in those. But when we look at the Alzheimer's disease brain, you can see coffee bean shaped nuclei. They look like coffee bean, and it looks like that the nucleus is actually folding, and that is the case. What is the mechanism of this? We know that lamin B1 is decreased in Alzheimer's disease and also in aging, and this contributes to excessive oxidative stress, but we don't know the mechanism of damage in here. The idea came from a great work by Bess Frost at Texas, and what she showed, she first time showed this coffee bean shaped nuclei in the Alzheimer's disease patients, comparing the normal nuclei here. And what do they believe? They believe that changes in tau causes misformation of microtubules. Microtubules will push down on the nuclear envelope, and then nuclear envelope will go inside the nucleus and will change the damage. And this is another image from Dr. Frost's lab, and she shows that this is a control, and the other nuclei is Alzheimer's disease nuclei, and you can see this deep invagination inside the nucleus. So what happens? What is the importance of this? We know that there are epigenetic changes. For example, methylation of autophagy. This is an important system for survival of neurons, and methylation of autophagy genes will cause less autophagy, and neurons are very sensitive, and they die. The other system is that we now know that nuclear invagination activates transposon. And for those of you in the field of genetics, you know that 45% of our DNA is non-human DNA, and most of those are transposons. You can have DNA transposon or RNA transposon, and what do? They are jumping genes. They can copy themselves and paste somewhere else in the genome. They can mutate genes, and they have been playing an important role in evolution, but they are also are dangers that are hidden in our cells, and if they wake up the way they do in Alzheimer's disease, now the hidden dragon will be awakened. So this could be an important part, an important avenue to study and try to find new drugs. And my lab examines the cause and the consequence of nuclear lamin damage. Here is a publication that I have cited here. This is evidence that these proteins and viruses are expressed in Alzheimer's disease, and these are all those transposons that I was talking about. So, oxidative stress is when there is an imbalance between reactive oxygen species and oxidants. So you need reactive oxygen species in normal healthy cells. If there is too much ROS, then you have cell and tissue damage. And we know that in Alzheimer's disease, this is happening because we know that there is too much protein oxidation, and also there is a decreased antioxidants. There are two major antioxidants, glutathione at the level of 2 to 10 millimolar, and thyroxine about 1,000 times less, and it's about 5 to 10 micromolar. And these are major antioxidants inside the cells. My interest in thyroxine system, and this is a protein here I'm showing you. Thyroxine has two thiol groups. These are two electrons that are easily extractable. And the importance of this is that the oxidized protein can easily come and take it. And this is a way that thyroxine controls the level of oxidized protein. And in this case, thyroxine will be oxidized. And if this thyroxine is oxidized, it's no longer active unless it's being activated by thyroxine reductase system. Inhibition of thyroxine system will increase the level of oxidized protein, and oxidized protein will induce cell death. So this is the work that Dr. Islam in my lab has performed. We were working on thyroxine system without any idea what's going on. And one day he came to me and he said, look at these nuclei. And you are looking at neuronal nuclei. We are stained for lamin P. You see that more or less beautiful staining on the edge of the nuclei. And then when we inhibit thyroxine, we see that the neurons, they shrink like a paper that is crumbled and you want to throw it out. And he told me, we have nuclear lamina damage. And my answer was, what is nuclear lamina? And then he educated me. So we first ran a Western blot, and we saw that after inhibition of thyroxine, we have a clip product of 46 kilodalton. He was trained in Alzheimer's disease, and he knew what this protein is. He told me that he thinks that there is a caspase-6 activation, and that's what we stained it for. And we saw that inhibition of thyroxine was associated with upregulation of caspase-6, and this seems to be causing nuclear shrinkage that we know. We tested this with measuring the level of procaspase as an indication of activation of caspase, and also measuring caspase activity in cell lysate. And we see that there is, with inhibition of thyroxine, we have a great deal of activation of caspase-6. So to give you a perspective, caspase-6, we know it is upregulated in Alzheimer's disease. So far, we didn't know what it is doing. And there are some groups that they are looking at caspase-6 inhibition as a biomarker for Alzheimer's disease. So this way, we see that in Alzheimer's disease, we know thyroxine is decreased, caspase-6 is activated. So we think that we have something here to contribute. We then used a caspase-6 inhibitor, and we showed you this nuclei that they were so damaged. Now they are perfectly fine if we inhibit caspase-6. And we show with Western blot that after inhibition of caspase-6 with the inhibitor, we can decrease the damage to a lot. So, and the question was that, is this also a glutathione dependent? Is this a general factor? When there is less antioxidants, you will have this damage? The answer is no, it is very thyroxine dependent. And in this experiment, we used 4 different reagents. BSO is a reagent that we use to inhibit glutathione. My apologies, glutathione only, and it doesn't touch thyroid system. And here we are looking at damage, and we can show you that only when we inhibit glutathione and no thyroxine, then the damage is not there. We confirmed that with measuring the level of caspase-6. We also measure it with amount of lamin B that is damaged, and we show that this is really just depending on thyroxine and doesn't have anything to do with glutathione. To confirm it more, we inhibited and downregulated with siRNA or shRNA and decreased the level of thyroxine reductase, and we see the same thing, and we confirm those massive nuclear invagination with these technique with electron microscopy. So now this is the part that is not published. We then use a human, we use a mouse that is widely used for Alzheimer's disease research and also in human brain autopsy sample. Here, this is the 46 kilodalton band that we showed you, and this is a new band at 21 kilodalton. This is again is happening only in mutant mice, and it doesn't happen in the control mice, or very much less is happening there. So I'm not going to reveal what it is today. These data are not published, and we are hopefully submitting it quite soon. We showed the same thing, that this enzyme is upregulated in human tissue, and we can show that 21 kilodalton band here again from the cleavage of this lamin B protein Alzheimer's disease patient, but not in controls. And we see a huge upregulation here. We're trying to show the upregulation of these enzymes in immunohistochemistry, and we can show that the signal, the green signal, is indicating that enzyme is hugely upregulated. Then we used a model of amyloid beta toxicity, and we showed that adding amyloid beta toxicity to neurons will induce cleavage of lamin B1 at 21 kilodalton. If we inhibit that inhibitor 1, we can completely inhibit that. Inhibitor 2 is also partly effective, so we can see that. We showed this the same thing with cortical mouse neuron, and we show it with immunohistochemistry. You can see these are the damaged nuclei, and then when we inhibit that enzyme, we can basically get away, and the number of neuronal damage decrease. We have techniques that we can actually measure the chromatin and changes in chromatin. I am moving towards gene regulation and changes. Here we are showing that this is a normal neuron and distribution of chromatin in white and empty spaces, in this case, in black. So here, that's the one. So these are the control panel. Adding amyloid beta, we can see that chromatin starts compacting. So you are losing a lot of genes, and you are gaining a lot of new genes that are now being accessible and transcribed. And with the inhibitor, we can recompensate by a lot, but still, I'm sure that this is not 100%. But anyway, you can see that there are significant changes after the inhibitors. We are looking at epigenetic markers here. We're looking at H3K9AC, which is a marker of genes that are being transcribed, and also H3K9Me2, which is a methylation marker, and this shows the compacting genes. We can see that with amyloid beta, the acetylation from normal cells changes a lot. Inhibitor number 1 can bring it back to a certain degree. Inhibitor 2 is okay. You know, there are some changes, but still we still have higher level. But we can see that these things can be actually reverted, and the same thing with methylation. So based on what I have told you, I want to say that there is hope for future, and hopefully there would be potential new therapies. As we talked about, we said that nuclear lamina damage results in nuclear envelope invagination that enhances retroviral expression. And now Dr. Frost's lab in Texas, they are pursuing antiviral therapy, and they are going to show if people that are on antiviral for a long time, would they have an added benefit of controlling the retroviral expression in Alzheimer's disease. They have shown it that it is very effective in a Drosophila model, but that remains to be done for human tissue, for human patients. And based on what we are proposing here, we do have some inhibitors that we can think of, and they could be alternative treatments for Alzheimer's disease by inhibition of nuclear lamina. For example, can we prevent thyroxine depletion? Can we use thyroxine mimicking reagents or caspase-6 inhibitor? In case of caspase-6 inhibitor, just recently it was reported that yes, adding caspase-6 inhibitor, at least to mouse that is overexpressing caspase-6, has improvement for memory and improvement in cognition. So the summary that I want to provide here is basically the hypothesis that we are following in our research. So we are thinking that amyloid beta toxicity and tau toxicity that are currently the target for treatments are actually byproducts of gene changes that is happening in Alzheimer's disease. We think that because of the oxidative stress, thyroxine level goes down. We know that inhibitor of thyroxine, TXC in thyroxine inhibiting protein goes up. We think that the second enzymes that we just mentioned is also upregulated. And all of these causes nuclear lamina damage by downregulating lamin B invagination. This is pathologic, and this causes acetylation and methylation are changes. Because of that, we have aberrant gene regulation, and they will be contributing perhaps to amyloid beta toxicity and tau toxicity. Eventually, all of these will lead to neuronal cell death. And I want to finish here. I hope I'm not over time. From University of Manitoba, these are my students in lab and collaborators from Canada and South Korea.